When we hear the word mitochondrion, the first idea that usually comes to mind is energy. That idea is correct, but it is incomplete. Mitochondria generate much of the ATP that powers the cell. They also provide metabolic intermediates that can be used to build lipids, proteins, and nucleic acids. And they participate in decisions that determine whether a cell survives or dies. So, in this section, we will follow one organelle through three linked roles: energy generation, intermediary metabolism, and cell death. Mitochondria have a bacterial history. They evolved from ancestral prokaryotes that were engulfed by primitive eukaryotic cells roughly 1.5 billion years ago. That history is still visible in the organelle today. Mitochondria contain their own DNA and use machinery that resembles bacterial systems for DNA replication, transcription, and translation. For example, mitochondrial protein synthesis begins with N-formyl methionine, and mitochondria can be affected by some antibacterial antibiotics. But the mitochondrial genome is only part of the story. Most mitochondrial proteins are encoded by nuclear DNA, while a smaller group is encoded by mitochondrial DNA. The mitochondrion is therefore built from two genetic sources: a small mitochondrial genome and the much larger nuclear genome. Because the ovum contributes most of the cytoplasmic organelles to the fertilized zygote, mitochondrial DNA is almost entirely maternally inherited. At the same time, proteins required by mitochondria can be encoded by either mitochondrial or nuclear DNA. That is why mitochondrial disorders can show maternal, autosomal, or X-linked patterns of inheritance. Mitochondria are not fixed structures that simply remain in place. They are constantly changing. Newly synthesized mitochondria can fuse with existing mitochondria, and mitochondria can divide through fission. These processes help renew the mitochondrial population and limit the accumulation of damage caused by ongoing oxygen-free radical injury. Mitochondria also have limited lifespans. Damaged or obsolete mitochondria can be removed by autophagy in a selective process called mitophagy. Depending on the tissue, nutritional state, metabolic demand, and injury, the estimated mitochondrial half-life is roughly 1 to 10 days. The important idea is that mitochondrial function depends not only on what an individual mitochondrion does, but also on how the population is renewed and quality controlled. The structure of a mitochondrion separates its major functions. It has an outer membrane and an inner membrane. The inner membrane is folded into cristae, which greatly increase the surface available for respiratory chain proteins. Inside the inner membrane is the matrix, the core compartment containing much of the organelle's metabolic machinery. Between the two membranes is the intermembrane space. The outer membrane contains porin channels that allow the passage of small molecules. Larger molecules, and even some smaller polar molecules, require specific transporters. This arrangement matters because energy generation depends on a difference between the matrix and the intermembrane space. The membranes are not just a boundary around the organelle. They are part of the mechanism. One brief note about the wording in the 10th edition of Robbins and Cotran that we are using. The matrix description mentions glycolytic and tricarboxylic acid cycle enzymes together. For compartment accuracy, glycolysis occurs in the cytosol, while the TCA cycle occurs in the mitochondrion. The major energy pathway begins when mitochondria oxidize substrates such as sugars, fatty acids, and other metabolic fuels. During oxidation, high-energy electrons are transferred from the original substrate toward molecular oxygen. The energy released along this process is used to pump hydrogen ions, or H+ ions, from the matrix into the intermembrane space. This creates an electrochemical gradient, more H+ outside the intermembrane than inside the matrix. The gradient stores potential energy. When H+ ions flow back down that gradient, the released energy is used to generate ATP. In this way, the mitochondrion converts chemical energy in substrates into a membrane gradient, and then converts that gradient into a usable cellular energy currency. The sequence is the point to remember: substrate oxidation, electron transfer, proton pumping, and ATP generation.
The electron transport process does not always have to remain coupled to ATP production. In brown fat, a protein called thermogenin, or uncoupling protein 1, can transport hydrogen ions across the inner membrane and dissipate the proton gradient. Substrates are still rapidly oxidized, but the gradient is no longer coupled to ATP synthesis. Instead, the energy is released as heat. This is non-shivering thermogenesis. The same process also helps explain why mitochondria can produce reactive oxygen species. Reactive oxygen species are a natural, usually low-level byproduct of substrate oxidation and electron transport. Hypoxia, toxic injury, and mitochondrial aging can increase this burden and produce oxidative stress.
Mitochondrial metabolism is not only about producing the largest possible amount of ATP. Robbins describes oxidative phosphorylation as an efficient way to generate energy from glucose and other substrates. The section gives an approximate yield of 36 to 38 ATP molecules per glucose molecule. But complete oxidation also reduces the substrate to carbon dioxide and water, leaving essentially no carbon moieties available for biosynthesis. Rapidly dividing cells have a different problem. They need energy, but they also need raw materials to make new lipids, amino acids, proteins, and nucleic acids. These cells increase their uptake of glucose and glutamine and may switch toward aerobic glycolysis even when oxygen is available. This is the Warburg effect. Each glucose molecule produces only 2 net ATP through this route, but the pathway preserves intermediates that can be redirected into biosynthetic pathways. So, mitochondrial metabolism can be adjusted toward 2 different priorities: maintaining cellular energy or supporting the growth and construction of a proliferating cell. Mitochondria are also central to cell injury. External insults, such as ischemia, toxins, and trauma, can damage the organelle. One consequence is the formation of mitochondrial permeability transition pores in the outer membrane. These pores dissipate the proton gradient. Once the gradient is lost, subsequent mitochondrial ATP generation becomes impossible. The cell loses the energy required for its essential functions, and this failure contributes to necrotic cell death. The causal chain is therefore injury, loss of membrane gradient control, failure of ATP generation, and loss of cell viability.
The apoptotic pathway is different. Apoptosis can be triggered by extrinsic signals, such as signals from cytotoxic T cells or inflammatory cytokines, and by intrinsic signals, such as DNA damage or intracellular stress. Mitochondria integrate pro-apoptotic and anti-apoptotic signals and produce a final decision to proceed or not proceed. When the net signal is pro-apoptotic, proteins including Bax and BAC form complexes that produce mitochondrial outer membrane permeabilization, or MOP. MOP releases cytochrome C and other proteins into the cytoplasm. Cytochrome C helps stabilize a multi-protein complex called the apoptosome. The apoptosome then promotes caspase activation, initiating the programmed cell death cascade. Here is the distinction that must remain clear. In necrosis, permeability transition dissipates the gradient and removes the cell's ability to generate ATP. In intrinsic apoptosis, MOP releases cytochrome C and activates the caspase pathway. Both involve mitochondrial damage or signaling, but they are not the same event.
The mitochondrial decision system has consequences for disease in both directions. If pro-apoptotic signaling fails, or anti-apoptotic effectors become excessive, cells that should die may survive. Robbins points out that this can support malignancy, even when mutations would normally trigger cellular suicide. The opposite imbalance can also be harmful. If apoptotic signaling is too strong, or anti-apoptotic protection is insufficient, cells may die prematurely. The section gives neurodegenerative disease as an example of the kind of problem that can result. Mitochondria therefore sit at a point where metabolism, injury, and cell fate converge. The mitochondrion is not simply a battery inside the cell. It is a dynamic organelle built from two genetic sources. Its two membranes and internal compartments create the conditions for oxidative phosphorylation. Its metabolism supplies both ATP and building blocks. Its fusion, fission, and mitophagy maintain the organelle population. And its membranes can become the site of either energy failure and necrosis, or cytochrome C release and apoptosis. The unifying idea is simple. Mitochondrial structure turns metabolism into information about the state of the cell. The organelle helps determine whether the cell has enough energy to function, enough material to grow, and enough damage to die.